Hi dear students, welcome you all to our biology class. Today we are going to discuss the most important and one of the difficult chapters of our plus 2 biology syllabus, that is molecular basis of inheritance. Okay, so today we are going to discuss an important experiment from this chapter, and this experiment is regarding DNA replication. So first we are going to discuss what is central dogma. Okay, so you might have heard the term central dogma. So what is central dogma? So central dogma was proposed by Francis Crick. The same Crick, in that Watson and Crick we have learnt, Watson and Crick proposed a double helical model of DNA, right? So that same Francis Crick proposed this central dogma also. So what is central dogma? We all know that proteins determine our various characters, right? So proteins are responsible for different characters in our body. So here we can see protein. So your any feature, for example, your height is determined by a protein, your hair color is determined by a protein. Okay, so first we all know that genes, we all have learned that genes are responsible for various characters in our body. Okay, so what are genes? We all know that genes are segments of DNA. Okay, so gene is a segment of DNA present in our chromosome. So this is our DNA. DNA is double-stranded. Okay, and the segment of DNA is called a gene. So double-stranded DNA. From this double-stranded DNA, first a single-stranded RNA will be formed, which is called as mRNA. We will discuss that in detail. So from DNA, a single-stranded RNA will be formed, and this RNA will give rise to protein. Okay, so DNA to RNA to protein. So we know that genetic information flows in this direction only. So genetic information flows from DNA to RNA to protein. Okay, so the formation of RNA from DNA, there is a term for that. It is called a transcription. What? Formation of RNA from DNA is called a transcription. Then what is the name given to the formation of protein from RNA? It is called translation. So RNA to protein is called a translation. So one more term is the formation of DNA from DNA itself. Okay, DNA is formed from DNA only. So that process is called a replication. So we have learned three processes: replication, transcription and translation. Okay, then in some cases, in most cases, RNA is formed from DNA and proteins, RNA gave rise to proteins. So the process by which RNA is converted into protein, we can say that this is an irreversible process. Okay, we cannot synthesize RNA from protein. This cannot be reversed. This process cannot be reversed. But here, in the process of transcription, that is DNA give rise to RNA. In some cases, for example, in case of some viruses, okay, in case of some viruses such as HIV virus, COVID-19 virus, etc. In case of some virus, their genetic material is RNA, and from RNA, what is synthesized? DNA is synthesized. Okay, from RNA, DNA is synthesized, and this process is called reverse transcription. Reverse transcription, and the enzyme responsible for this reverse transcription is reverse transcriptase. Reverse transcriptase. Okay, so this is central dogma. According to central dogma, DNA is formed from RNA is formed from DNA, and this RNA later give rise to protein. Okay, so the process involved are DNA to DNA, DNA formed from DNA, the process is called replication. RNA formed from DNA, the process is called transcription. Protein formed from RNA, the process is called translation. Then formation of DNA from RNA, it is called a reverse transcription. It occurs in some viruses, and the enzymes responsible for this process is reverse transcriptase. Okay, then next we are going to discuss about DNA replication. Okay, so what do you mean by DNA replication? DNA, the formation of DNA from parental DNA. Okay, copying of DNA from parental DNA, it is called DNA replication. So in DNA replication, the most important thing we have to learn is that DNA replication is semi-conservative. Very, very important thing. So DNA replication is semi-conservative. DNA replication is semi-conservative. So what do you mean by semi-conservative replication? So here you can see DNA has got two strands. Okay, one strand and the second strand. Okay, so this is our parental DNA. So when this DNA undergoes replication, first the two strands will separate. Okay, the two parental strands will separate. Okay, both the strands got separate. Then each strand will form its own complementary strand. Okay, each strand will give rise to its own complementary strand. So I will show this thing with the help of sequences also. So okay, here this is one strand, and we are giving some nitrogen bases or sequences here, A, T, G, C, A, G, A, T. Okay, then what will be the sequence of this opposite strand? T, A, C, G, T, C, T, A. So as we all know, adenine always pairs with thymine and guanine always pairs with cytosine. Right, isn't it? Okay, then these two strands get separate. Okay, this is one strand and this is the second strand. So in this strand we can write A, T, G, C, A, G, A, T. Okay, then in the second strand sequences are T, A, C, G, T, C, T. Okay, so we have already said that this strand will synthesize its own complementary strand. Its own complementary strand means a new strand will be synthesized. Opposite to A, T will be there. Then A, here C, G, T, C, T, A. So this will be one new DNA. Then in the second strand also, what will be the sequences opposite to T? A. Here T, G, C, A, G, A, T. Okay, so we have got two DNA, right? From one parental DNA, two daughter DNAs are formed. So here we can notice one thing. One strand is from the parent. Okay, so this one is the parental strand. So in both this DNA we can see a parental strand, right? So one strand will be a parental strand and the other strand will be a newly synthesized strand. So one will be a parental strand and the other will be a newly synthesized strand. Newly synthesized strand. Okay, so here we can say that DNA replication is semi-conservative. Why DNA replication is semi-conservative? Because In a newly formed DNA, one strand will be from the parent. That is, one strand will be parental strand and the other strand will be a newly synthesized strand. As one strand is from parent, it is semi-conservative. If both the strands are from parent itself, it can be called as fully conservative, right? But here only one strand is from parent and it is called a semi-conservative replication. Okay, so the experimental proof for semi-conservative replication, it is called a Michelson-Stahl experiment, and this experiment we will discuss in our next class. Thank you.